And also the earning of the people who have in the bachelor diploma is also higher than the people without a bachelor diploma. You can see here, this is the table in the right is the life earning, which means that the money they earn in a lifetime years of serving at their position at the moment. You can see that if the people who have been graduated, he can make more than $2 million in their lifetime, but the people without bachelor diploma just earn about more than a half of that money. It's just about 1,300,000. But there is one more information is quite interesting. That is, as you can read on the title here, that only 27% of the college graduates have the job related to their major. That means that nearly 3 of the quarter of you will have the job not related anything with what you are studying at university. So what does that mean? Does that mean that all the knowledge you obtain in university is useless? Then why you have to go to university? Because you go to university, you have time consuming, because you will lose around 3 or sometimes 4 or 5 years of your youth, which is a very valuable property. Youth is the most valuable property of each person. And also university is also costly, because it costs a large money of your family, your parents, to go to university. So why we have to go to university, since a large proportion of you doesn't need it anyway? It's quite a tricky question, interesting question. In my idea, so if we think that the university program is also useless, why statistics numbers showing that the people who graduate from university have a high chance and also higher earnings than the people without a bachelor? Okay, I'm saying in general. I didn't want to mention about some very critical, exceptional, like Bill Gates or Steve Jobs, who drop out their university but still have a very successful life. So anyway, I think the most important thing in university that you study is not just about the knowledge you have, but also the most important property is adaptation. Because when you graduate, you don't know what career you will develop in. You never know what life throws into you, right? Because as you can see here, 27% of college graduate, only 27% of the graduate have the job related to their major. That means, among you, 3 will have the job doesn't relate at all. That means that we never know what kind of job we will obtain in the future. Maybe you graduate from the ICT department, but you finally get some job in a bank. Not like the IT guy in the bank is still related to your job, but like a banker, like a credit guys who take credit from customer, call the customer for loans, something like that. So what is the most important thing is adaptation. What can you really adapt with your new position, your new career, and the new field that you have no knowledge, no experience in. So that's why in university, in the first year, you have to cope with different subjects where you are not familiar with. So you can try to handle those difficulties by your own ability. Okay, so by on that you will have a little proud. Oh, in the past I have been cope with a situation like this. I have to work in the field I know nothing about, but I still overcome it, and I know how to overcome it, and that adaptation will be helpful for you in the future. Okay? All right, so in Monday, we have discussed about from the past to nowadays modern genetics, what does it mean, and what is the application on genetics. We know that nowadays we have a program that from your genome, it can predict from your genetic background what is the proportion you get a certain disease in the future, like type 2 diabetes, like cancer. It can also predict the baby who has not been born yet from the embryo stage. We still know that which kind of disease this baby will have in the future. We also can predict the age, the height, not very precisely, but accurate enough. The skin color, the eye color, and all related, all the physical appearance of those individuals just based on the genetic background. And in last week, we have seen Italian scientists present about their AI program just to predict the facial feature of an individual just based on their genomes. That's quite interesting. So where can they extract those information? So first, let's come to what is the structure of a DNA. Okay. So DNA is stand for deoxyribonucleic acid, and is composed of three main components. First is a sugar, a 5-carbon sugar called a deoxyribose sugar, a phosphate group, and a base nitrate. So this is all the components form a nucleotide. Wait a minute, draw. So a DNA is a polymer of a single unit called nucleotide. So a nucleotide is composed A nucleotide is composed of three main components. First is a 5-carbon sugar. Called deoxyribose sugar. A phosphate group and base nitric. So with the 5 common sugar in the, so we have a oxygen here, and we have 5 carbon. There's no sound. Everyone, you can hear me? Because there are some students heard there's no sound.
Yeah.
Yes.
Yes, we can.
Yes, we can hear you.
Okay, so what is the problem with this student? Okay, so we have a 5 carbon sugar here. We have carbon number 1. So remember that right in the right of the oxygen atom, we have the carbon number 1. Carbon number 1. Carbon number 2, number 3, 4, and this is carbon number 5. Okay. So this is a 5 carbon sugar deoxyribose. What is the differences between the deoxyribose and the ribose? I will explain later. But first, let's focus on the deoxyribose sugar. So we will have a phosphate group attacked into the carbon number 5 here. This is a phosphate group. So we have all this is a 5 carbon sugar. This is a phosphate group. And we have a base nitric attacked in the carbon number 1 here. So this is this is the base nitric. So this is the 3 component of a nucleotide. Okay. So normally nucleotide, because of the phosphate group, will have a negative charge at the neutral pH. So this property is quite important when we want to electrophoresis. That is a technique to identify the size of the DNA fragment based on the distance it travels when we put it in electricity. We will come to this technique later. But the difference between the ribose sugar and the deoxyribose sugar is in the carbon number 2 in here. Okay. In the carbon number 2 in here. So in the ribose sugar, it's a hydroxyl group, and instead of the hydrogen atom in the deoxyribose. So in here, carbon number 2. This, I will change the color. Color is green. So this position in here is the OH, the hydroxyl group. Or called hydroxyl. In ribose sugar. And this is the hydrogen atom without oxygen. in deoxyribose group. So the term deoxys, that means without oxygen, okay? So the differences between the ribose and the deoxyribose is in the carbon number 2. Instead of the hydrogen in the deoxyribose, we will have the hydrogen group in the ribose. Two nucleotides combined together, it will have a covalent link between the carbon, the hydrogen group of carbon number 3 of the first nucleotide here. It will combine to the phosphate group of the carbon number 5 of the next nucleotide, and the result it causes a phosphate diester link here. This is the covalent link. That's why it's quite stable. Okay. So this link is formed from the carbon number 3 of the first nucleotide to the phosphate group and to the carbon number 5 of the next nucleotide. And that's why we call the direction from the up position to the down position here is the 5' to 3' end. So please pay attention at this part. In here, from this direction, okay, is called the 5' to 3' direction. Because in this direction, the carbon number 5 comes first and the carbon number 3 come after. Okay. So like we have the nucleotide here. And now this nucleotide is, let me to remove all this. So if we have another nucleotide and we will have a covalent link between the carbon number 3 here to the phosphate and of the carbon number 5 of the next nucleotide. So this is the phosphodiester link, and also is a covalent link. And because of this link, we can see that the direction from up to down is the 5'. This is the 5' to the 3' direction, because the upper is the 5 carbon come first and the 3 carbon come later. Okay, remember this because you will use this information to handle the exercise. Okay, so you have to understand what is the 5' end of DNA strand and what is the 3' of the DNA strand. This information are also important in the next chapter in DNA replication and DNA transcription, because you will know that this type 2 process, both DNA replication and transcription, only happen from one direction, not the other peoples. Okay. So for the nucleotide, there are 4 types of nucleotide in DNA depend on the base matrix. So in the composed of a nucleotide, we have a 5-carbon sugar, a phosphate group. This two is identical. These 2 parts are identical. In all nucleotides. The only difference is this part. This is the only difference between nucleotides. So we have 4 types of base matrix, which is A, stand for adenine, T stand for thymine, C stand for cytosine, and G stand for guanine. So this is 4 type base matrix, and because of this, there are 4 type base matrix. So we have also 4 type So for this, we will have this cause also of nucleotide. Which is ACGNT. And G, follow the name of the base matrix. So that's in DNA. In RNA, we will have a U, which stands for uracil. Okay. In RNA, we have So in RNA, we will have the RNA will have only A, U, G, and C, not A, T, G, and C. And the structure of the base is, as you can see in the slide here, the A and G is two big structure of base matrix. So it was categorized as purine. So we have see that the structure is quite similar between the A and the G here. And there are 3 base matrix, T, thymine, uracil, and cytosine is the small one is called as primitives. Okay, and the structure of T and U are quite really similar. It's the only differences in here is the position here. You can see that in the this position, the position 1 here, the methyl group was attacked in the position number 1. Instead in uracil, we have only the hydrogen atom. Okay. And based on the hydrogen bond, the A is always linked to T in DNA, and A is always linked to U in RNA by 2 hydrogen bond, and G is always linked to C by 3 hydrogen bond. The position of the hydrogen bond between A, T, G, and C are placed in the slide. So you see here is the 2 hydrogen bond between thymine and adenine, and the other is cytogens is linked with guanine by 3 hydrogen bonds. So because C is linked with G by 3 hydrogen bonds, so normally the 3 bond will be more stable than the 2 hydrogen bond. So that's why with the fragment of DNA that rich CG, we will have normally it with more stable than the fragment with more ANT. So that we need more temperature, more energy to break down, to denaturate the fragment of DNA with rich CG.
Excuse me. So you mean that the A and like the DNA with more T and C are more stable than DNA with A and T?
Exactly. Yes. So in the chapter practical, we will know about a technique called Pcr. Okay, and the Pcr is a technique that you use it to detect the COVID-19, right? So do any one of you have heard about this technique? It's quite famous recently based on the detection of the COVID-19. So the Pcr is built on first we have to denaturate the DNA fragment. So based on the DNA fragment separated, it will be more amplified based on the sequence. Okay, so with the DNA fragment with more CNG, we call it rich CG. Normally the temperature and also the energy to break down the DNA fragment is much higher than the fragment with rich ANT. So we will need more energy, we'll need more temperature to break down the fragment of DNA with rich CG. We'll come about it later when we come to the practical chapter. I don't know, maybe we can go right now. Wait for me a minute because of the question. Practical chapter. Yeah. Okay, so I will explain very quickly about the Pcr technique and use the. So first, let's watch a small clip to demonstration what is the technique is. This is very useful technique when we want it to detect any sequences. For example, if we want to detect that in a sample, if the patient was infected by COVID-19 or not, we will insert in there we will detect a specific sequence of the COVID-19 because we have already sequenced all the genome of the COVID-19. So we will use this technique to detect that in the sample there's any DNA fragment of the virus. Okay. First, let's.
Polymerase.
Wait for me a minute.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Okay, so I will explain. Có lẽ là cái đoạn này mình giải thích luôn ở tiếng Việt cho nó nhanh, bởi vì là kiến thức này là kiến thức bổ sung thêm thôi, chứ nó không phải là kiến thức chính. Là cái kỹ thuật PCR là gì? Thì kỹ thuật PCR thực ra nó là một kỹ thuật cổ điển trong sinh học phân tử, dùng để phát hiện những cái đoạn DNA mà mình đã biết trước trình tự. Và ứng dụng của nó, ví dụ như là mình muốn phát hiện con COVID-19 trong mẫu bệnh nhân, mẫu có thể là mẫu nước bọt hoặc là mẫu dịch mũi, dịch nhầy, thì đầu tiên là mình cần giải trình tự toàn bộ cái con COVID-19 đấy. Thì các nhà khoa học trên thế giới đã làm rồi, code trình tự gen lên rồi. Thế thì mình sẽ target một đoạn trình tự nào mà chỉ có con virus này có mà các con virus khác không có, được chưa? Sau khi target rồi thì mình sẽ thiết kế một đoạn một để nhân cái đoạn gen đấy lên. Thì cái kỹ thuật PCR này là để nhân cái đoạn gen đấy lên, thì nó gồm 3 bước. Bước đầu tiên là denaturate, có nghĩa là máy nó sẽ đẩy cái nhiệt độ của cái đoạn DNA này lên khoảng độ 95 độ C. Thường mình chạy là 95 độ C. Thì ở 95 độ C thì hai cái đoạn DNA nó sẽ tách nhau ra. Tuy nhiên như lúc nãy mình giải thích là thông thường những đoạn DNA nào ấy mà có cái nhiệt độ, mà có cái đoạn GC chứa GC nhiều, thì bởi vì G với C liên kết với nhau bằng 3 liên kết hydro, cho nên nó bền hơn khá là nhiều so với cả cái liên kết giữa A với T thì nó chỉ có 2 liên kết hydro thôi, 3 thì bao giờ bền hơn 2, đúng không? Thì cái nhiệt độ tùy thuộc vào cái phần trăm GC mà cái nhiệt độ này phải tăng lên, vì là nếu liên kết giữa hai đoạn mạch DNA mà nó bền thì sẽ cần nhiều năng lượng hơn để mà có thể phá vỡ được cái liên kết giữa hai mạch ADN đấy. Thế thì các đoạn mồi sẽ gắn vào đây và máy lúc này sẽ đẩy về cái nhiệt độ khoảng độ nhiệt độ bắt mồi khoảng độ 68 độ C. Thì đoạn mồi sẽ gắn vào đây và DNA polymerase bắt đầu làm việc để nhân cái đoạn gen nó lên. Và sau đó thì cái nhiệt độ này lại đẩy lên 72 độ C để mà cái đoạn DNA này có thể được tổng hợp. Thế thì đấy là một chu kỳ, và thường một phần PCR sẽ diễn ra trong vòng từ 30 đến 40 chu kỳ. Có nghĩa là sau hết một chu kỳ thì nó lại quay lại cái vòng này, lại đẩy trên lên 95 độ C, xong lại quay về nhiệt độ bắt mồi ở 68 độ C, xong 72 độ C, xong lại hết một chu kỳ lại quay lại 95 độ C một lần nữa. Thì cứ sau một chu kỳ là số phân tử ADN sẽ được nhân lên 2 lần. Sau một chu kỳ là số phân tử DNA sẽ tăng lên gấp đôi, 2 chu kỳ để tăng lên 4 lần, 3 chu kỳ sẽ tăng lên 8 lần, 4 chu kỳ sẽ tăng lên 16 lần, như thế. Thì cái đoạn DNA đích của mình sẽ được nhân lên, và nhờ cái đoạn DNA đích nhân lên đấy mình biết là cái phân tử DNA mình cần, cái trình tự DNA mình cần là có trong mẫu hay không. Cái bệnh nhân nào mà âm tính có nghĩa là trong cái mẫu của bệnh nhân đấy là không có vết của virus, còn bệnh nhân nào mà dương tính cũng là xuất hiện vạch thì bắt đầu có cái DNA của virus ở trong này, có thể là tức là có thể là bệnh nhân là bị nhiễm rồi, đúng không? Has there been a case where human DNA is mistakenly target? Mistakenly target nghĩa là sao? Nghĩa là nhận diện nhầm đoạn DNA của con người, đúng không? Mistakenly có nghĩa là sao? Ok, ở đây thì cái việc nhân cái đoạn gen này lên thì khi mình muốn biết là trong cái mẫu của mình có con COVID-19 hay không, thì mình sẽ chỉ nhắm đến cái đoạn gen nào mà chỉ cái con đấy có, các con virus khác ví dụ con cúm bình thường không có. Đấy để người ta phân biệt giữa bệnh nhân nhiễm COVID-19 với cả bệnh nhân mình nhiễm cúm thông thường, đúng không? Thì người ta phải nhắm đến cái đoạn gen nào mà chỉ con này nó có, con COVID-19 này nó có mà các con khác không có. Thế thì con khác không có thì người cũng không có, chỉ có mỗi con virus COVID-19 thì có thôi, thì cái khả năng để bắt nhầm đấy nó rất là ít, được chưa? Okay. So we will come to a new term, which is a complementary strand. So what is the complementary strand? Okay, so normally DNA fragment have two strands, and we will have we can see here I will use my pen again. I will use this copy. Okay, so in the DNA double strand, so this is the DNA double strand. Okay. So if we have one, so this is let's zoom out one DNA fragment. So we have a DNA strand here, which is from the 5' to 3'. This is the 5'. And this is the 3'. Let's say that this is A adenine, and this is the G, okay? Let's say that this is A, and this is G. So in the other strand in here, we will have because in the other strand, it will have the converse complementary. That means that in here, we have, so now the carbon will add it number 1 here, number 2, number 3, number 4, and number 5 here, right? So in here we will have, because this is an A, so this might be a T, linked with each other by 2 hydrogen bonds. And we have another nucleotide will link the same with this. So because this is a G, we will have this is a C. With 3 hydrogen bonds. So as a result, you can see here, what is this direction? The direction is here is 3' to 5', right? Because the 3' come first and the 5' come later. So this is the 3' and this is the 5'. So in a DNA double strand, if one strand is from the 5' to 3', then the other must come from 3' to 5', right? So this is what we call a complementary strand or an anti-parallel strand. Because if this one from 5' to 3', then the other must come from 3' to 5'. So let's focus on this example. So in here I have a fragment of DNA. So in here I have a fragment of DNA. Let's overcome the exercise on the protein because we have not studied the transcription and translation yet. But just focus on the direction, 5' to 3'. So in here, this DNA is the 5' to 3'. Then we can see that this one is from 3' to 5'. With the sequence of nucleotide is follow the complementary rule, or we call it the Jagat rule, that A is always linked to C, and G is always linked to C. So this is a reverse strand of the first strand here. And if we want to rewrite this one from the 5' to 3', so then we will have from this direction to this direction. So we want to reverse the 5' here, C, T, A, G, 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 T, G, A. So C, T, A, G, G, T, G, A, and T, G, C. Okay. So this is how the negative strand or the reverse strand of the first strand that we write here should write, because all the DNA should be written from the 5' to 3', because later we will discuss all the DNA replication and also all the transcription will happen from the 5' to 3', not the way around. Okay. So we will come back to this direction again on the chapter 2 and chapter 3. Now, now we have a small exercise for you. Please spend 5 minutes and handle the exercise for me. So the exercise here is a scientific design primer, which is a short oligonucleotide for use in a DNA sequencing reaction. The primer is complementary to the DNA template here. So this is the DNA template, which written from the 3' to 5'. So please write down for me the primer, that means the complementary strand of this DNA fragment from the 5' to 3' direction. So to save time for you, maybe I will put it in the chat box. So this is, I put in the chat box for you the sequence of the DNA fragment. Can anyone write down for me the sequence of the primer? Yes, you can write down directly in the chat box. Or if you have any question. Yeah, please write in the chat box. Could you help me to send the solution for everyone, please? Just click on the tool and send to everyone in the meeting. It will be better because all the class can see your solution. Yes, thank you very much. So we have a slightly different, yeah. So correct, most of you have the correct answer. So the exact solution is TCGA. So we can see here. So we can see here because of the complementary rule, A is always linked to T and G is always linked to C. So, and this is already a parallel DNA strand. So we will come that the A is always linked to T. I don't know if you can see my mouse moving. Anyone can see the pointer? The
Yes.
mouse?
Yes. Okay.
Yeah. Okay. So the G is always linked to C. So we can rewrite the primer of this DNA fragment as so A is T here, G is C, C is G, T is A, and A is T. Okay. Okay. Wait. So now we will have a small break. I'll say a 5-minute break, and next we will come to the differences between DNA structure and RNA structure. Okay, 5-minute break. Okay, so let's come back to our lecture. So the main difference between the DNA and RNA structure is in DNA, most of the DNA strands is the double strand. And the composed of the sugar in the nucleotide structure. In DNA, nucleotide structure was composed by a deoxyribose. Meanwhile, in RNA, the nucleotide was composed by the ribose sugar. Another differences is between the type of nucleotide. In DNA, we have thymine, and in RNA, we have uracil. Okay, so for more understand more about the structure of DNA, let's watch a small, animation.
Do you recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT, or TA, and GC or CG base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms two repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair non-specific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks. This is the DNA double helix, and you've now learned the structural features that influence its function. We hope you've enjoyed exploring this amazing molecule with us.
Okay, so coming up next is one of the important information that if you still remember about the survival kit. So if you remember about the survival kit for the student who just want a minimum result, just to pass the course. So today we have studied two things you should remember. First is the structure of nucleotide. Okay, what is structure of nucleotide? You have already know it's composed of three main components: sugar, 5-carbon sugar, deoxyribose in DNA and ribose in RNA. Phosphate group and base nitric. Okay, and the second part is structure of DNA and RNA and the difference between the structure of DNA and RNA. So the part is I have already made a here. Differences between DNA and RNA. Okay, so the main differences between DNA and RNA, first in the structure of sugar, DNA is composed by a deoxyribose sugar and the RNA is composed by a ribose sugar. The type base in DNA we have HCG and T, and in RNA we have HCG and U. Thymine in DNA was replaced by uracil in RNA. Okay. With the structure strand in DNA, most of the DNA molecule is a double strand. That means it's composed by two DNA strands in parallel. One is from 5' to 2', and the other is from 3' to 5'. While most of the RNA is single strand, where some RNA is double strand in some genome of the virus. We will come about it later, but most of the RNA molecule is single strand. DNA is the genetic material of most living organisms. Eukaryotic cell, prokaryotic cell is DNA, and in some A large proportion of the virus is also used DNA as its genetic material. In RNA is using as a genetic material for only some kind of virus. We will come to that virus in a few slides. The function of DNA is to replicate. We will study about DNA replication in next Monday. Store the genetic information to passing through to the protein and also passing through to the offspring. Meanwhile, in RNA, we have mRNA is transfers the genetic information from DNA to protein. We have also other structured RNA like ribosome RNA or non-coding RNA, but we will come about the other type RNA later. We have also tRNA, ribosome RNA, long non-coding RNA, short non-coding RNA, many types of the RNA other than mRNA. About structure, DNA is more stable because it have composed by 2 DNA strands, and because of the complementary bond between the 2 strands, make the molecule of DNA is more stable. Meanwhile, the RNA is less stable. It's less, much less stable than DNA because first it's a single strand, most of it. And second, the hydrogen group in the ribose sugar is more susceptible to hydrolysis, which is to cleavage the structure of the ribose by hydrogen reaction. And finally, is the length. The DNA, normally a DNA is much longer than RNA molecule. Okay. So next, come to the genome of the virus, a prokaryotic cell and a eukaryotic cell. So the virus normally is quite have a very simple structure. It's so simple that even some nowadays many scientists are not consider a virus as a living organism, because according to them, a living organism must have a specific characteristic, is a metabolic mechanism. In virus, they don't have the metabolic mechanism. They cannot synthesize energy by themselves, but mostly based on the host system. The structure of the virus is very simple. It's composed of one nuclear core. The nuclear core can be DNA, can be RNA. The nuclear core can compose by single strand or double strand. And the envelope was composed of protein. Okay. So in virus, the genome of the virus, the genome can be both single strand or double strand, DNA or RNA. The genome can be linear or circular. That linear, that means that it's one DNA molecule have the end and the beginning, and in circular is composed like a circle molecule with no ends. Normally, the DNA virus, we have genomes much more stable than the RNA virus. So because of the simple structure, normally the genome of the virus is quite small. The size is from 2,000 to millions base pair. To compare, the genome of a human being is about 3 billion base pair. 3 billion. So this is the structure of 2 typical RNA virus. The HIV virus, which calls the AIDS disease AIDS, and the coronavirus, which is causing the COVID-19 pandemic that we still have to suffer heavily nowadays. So let's take an example on the infectious pathway of the HIV virus. So in the case of HIV virus, it will infect only on the T4 leukocyte in the immune system of human. So it's a T4 white blood cell. So it recognizes the T4 white blood cell based on receptor called the CD4 receptor on the membrane of the T4. So the HIV in the protein envelope of HIV is have also its receptor called the GP120, and when it reach the CD4 receptor of the T4 cell, it will cleavage the envelope protein, activate its infectious pathway. First, it will inject its RNA nucleocide into the host system, and this RNA will using the reverse transcription is the enzyme that convert the RNA strand to the DNA strand. So from this RNA, based on the enzyme reverse transcriptase, it will synthesize the cDNA of the virus. This DNA will match into the nucleus and match also in the DNA nucleocide of the host, which is the T4 leukocyte cell. And it will use the system of the material or the system of the host cell to synthesize the new DNA, the new RNA, and from this RNA, the transcription started to synthesize the protein envelope, and from one single copy in the beginning, it can be multiply 1,000 times to make another 1,000 other virus to inject into the other cell. So next come to the probiotic genomes. The probiotic genome is firstly is DNA. We only have virus is the only cell that have RNA as a genetic material. In all the prokaryotic and eukaryotic, we have DNA as a genetic material. So first, the genome of prokaryotic cell is DNA. And normally it's one circular chromosome. That means it's a circular DNA. It's packed quite dense in the nucleus region, like in the red region here. The circular DNA of the prokaryotic nucleus cell is packed in here, and in many prokaryotic cells, it have its own plasmid. It's a DNA string. It's from a DNA string, small string circular. Normally, it will have 1,000 of version of plasmid, and this plasmid is composed in a role to against the antibacterial substances that we will study later. DNA, it have different shape. First is normally DNA is in the stress, right? Relaxed DNA. Relaxed DNA in the that means the DNA genomes was circular DNA genome is in the relaxed shape here, but it only in the relaxed shape when the DNA need to replicate or transcribed. Sorry. Normally, it will appear under the supercoil shape. That means the DNA fragment, DNA molecule was twisted in the maximum dense phase like this to easier to preserve and easier to separate during the cell division. Of course, under the supercoiled state, none of the DNA replication enzymes or transcription system enzyme can attach into the DNA to start DNA replicate or DNA transcribe. Because in chapter 2 and chapter 3 we see that to launch DNA replication or RNA transcription, we will have, in case of DNA, is DNA polymerase system complex. In case of RNA transcribe, it's transcription factor complex. It is a huge it's a combination of different enzymes, and it will take a lot of space. So under supercoiled phase, there's no much space for this system to come and binding into the DNA strand. So the process cannot happen normally. So this is a result of electrophoresis of the DNA plasmid system under the different shape. So we can see that although the DNA plasmid have the same side, we can see that the supercoil, the dense one here, is moves in the fastest. The liner is moving less fast, and the slowest is the nickel circle.
The circular chromosomal DNA of bacteria must be compacted about 1,000-fold to fit within a bacterial cell. Part of this compaction comes through the formation of loop domains, where segments of DNA are folded into loop-like structures that are held in place by DNA-binding proteins. The formation of loop domains compacts the DNA about 10-fold. Further compaction is accomplished through a process known as supercoiling. In this process, which is under the control of the enzymes DNA gyrase and topoisomerase 1, twists are introduced in the DNA molecule that cause it to compact by coiling onto itself. This is much like the process of coiling that you can observe by twisting a rubber band. DNA looping and supercoiling make the bacterial chromosome more compact so that it can fit within the nucleoid of the bacterial cell.
So in the eukaryotic cell, we will see that in the eukaryotic cell will have the same system to make the DNA was more protective and more easier to maintain its function and more stable. So come to the eukaryotic genomes. The eukaryotic genomes was located in the nucleus of the cell. So this you have studied in cell subject. And in many cells, beside of the nuclear genome, which is located inside the nucleus of the cell, many cells will have we call a second genome in the mitochondria and in the chloroplast in the case of the plant cell. So different with the prokaryotic genomes, the eukaryotic genome is a linear double strand, not a circular, okay? A linear double strand. And in eukaryotic cell, the genome is arranged not as a single molecule, but as several molecules, and each molecule of DNA is attached with a protein called histone. And this protein histone, the complex between the protein histone, the DNA molecule, will compact into the structure called chromosome. So this is the structure of the complex of DNA and nucleosome. So we will have the DNA. Let's compose that this is a double-strand DNA. This double-strand DNA was composed not alone, but composed with a protein histone. So this is a protein histone. We have H subunit like this. So this is and this is one unit code like this. The DNA will wrap around one this will wrap 1/3 around the nucleosome. And a nucleosome is composed of 8 subunits of protein histone. Two subunits of H2A, two 8 unit H2B, two 8 unit subunit of H4, and two subunit of H4. And the subunit H1 is in here. So this is the H1. This H1, or sometimes we call it linker histone or linker protein histone. Okay. So we can see in here the DNA is wrapped around 1/3 aside of the nucleosome. Each nucleosome will compact here to form a chromosome, and the protein histone H1 is the linker between two nucleosomes. And under the microscopic imaging, you can see the nucleosome was organized like this. So let's watch a small clip on the structure of a nucleus.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So you see that here a nucleosome was composed by 8 subunits of histone, 2A2A, 2A2B, 2A3, and 2A4. And in each structure, we will have a part of the protein was put inside. Part of the protein, a subunit of histone was put inside. This is called histone tail. This histone tail play a critical, very important role in the epigenetic that we will study in chapter 7, but we will come to that later. And DNA wrapped in here in these 6 nucleotides, and the histone 1 is played as a linker. And the package in the have been twist quite dense in a different level depend on the period of time in the cell cycle. Normally, the chromosome was observed most clearly in the metaphase of the mitotic cycle. So we come to 2 terms in a state of the chromosome is heterochromatin and acrochromatin. The heterochromatin is the shape that the chromosome was densely twisted in the shape here. We can see in like this. And in this shape, normally, none of the in the prokaryotic cell in this shape, when it's in the state of magnets, none of the transcription factor nor the DNA polymerase can come to attack and start to replicate the DNA or transcribe DNA. To maintain these 2 processes, the heterochromatin chromosome must transfer into another stage called acrochromatin, which is more open and less dense. So the chromatin was twisted out to make space so the transcription factor and also the replication system can have the space to combine and start to replicate and transcribe the DNA. Okay, so the next part is, okay, so we will come to the last part of today is the Endosymbiotic Theory. So as we have discussed a few minutes ago, inside a genome, beside of the genome that located in the nucleus of the cell, here in the term of animal cell, and here in the term of plant cell, we will have a second genome located in the mitochondria here. And in terms of the plant cell, both mitochondria and the chloroplast here. So scientists find out that in all the organelles of the cell, the mitochondria and the chloroplast have its own DNA. And this DNA, when they sequence this, the DNA sequence of this genome, they find out that it's quite similar to the prokaryotic cell. So they have made a hypothesis called the endosymbiotic. That means in the ancient time, let's say around, I don't know when exactly, but more than billions of years ago, somehow there's a symbiotic between prokaryotic cell and the eukaryotic cell. That some prokaryotic cell have come and binding inside eukaryotic cell to maintain its function as energy center of the cell. That's what a small clip on the endosymbiotic theory.
Endosymbiotic theory explains how scientists believe eukaryotic cells developed. First, the nucleolus was surrounded by the enfolding membrane, creating the nucleus. Then further enfolding membrane formed the endoplasmic reticulum. Then the cell engulfed an aerobic bacteria, which would ultimately become the mitochondria. Afterward, a cell with mitochondria engulfed a photosynthetic bacteria. This photosynthetic bacteria would become the organelle known as a chloroplast. Mitochondria and chloroplasts contain their own small supply of DNA, which may be remnants of the genome they had when they were independent bacteria. It is important to note that we know that aerobic bacteria were engulfed first because there are animal cells which lack chloroplasts, while both plant and animal cells contain mitochondria.
Okay, so the next part on the human mitochondria genome is quite long, so I think I will save it for next Monday. So the today lecture is finished here. Do you have any question or comment? Okay, so since you have no more question or comment, I thank you very much for your attention.
Wait.
Yeah.
I'm sorry. I want to ask about our practical session next week.
Practical session, okay. Hello, you hear me?
Yes.
Okay.
Yes. So do we go to the university to study that lessons, or will we continue on studying online?
Well, as the schedule, the GAD, the General Education Department, sent to me, we still have an online practical. So you can stay at your home and remember to access into the Zoom. I believe it's still a Zoom class. Yeah, into Zoom online on time, and we will start our practical online. Okay.
Okay, thank you.
Okay, thank you very much, and see you next Monday.
Goodbye, teacher.
Goodbye,
See you.
Goodbye, teacher.
See you.
Goodbye, teacher.
Bye, teacher.